Hi, everyone. Thank you for signing up for this webinar. I hope it will be very useful to you. It's quite exciting stuff going on. So I am actually joined by my colleagues Ben and Alina, who are also part of the Ensembl outreach team, and Francesca, who is part of the GeneBuild team, so the team who makes the gene annotation for our data. So thank you, guys, my colleagues, for being here, for answering any questions in the background. So if you have any questions at all, please feel free to add them in the Q&A. My colleagues will answer them in the background, and I'll also try and answer some live example, a question about going through an example again. So before I start, I want to show you what I'm trying to achieve for you at the end of the webinar. So by the end of the webinar, I hope that you will be able to describe what the Human Pangenome Reference Consortium is, or HPRC, and how Ensembl is involved. And most importantly, how to actually retrieve annotated genomic data via the Ensembl Rapid Release website. So all of my slides will be available on our training page. So I'm just going to show you how this looks like. If you open your browser and go to training.ensembl.org, so I'm going to do this on my screen right now. I've switched to Google Chrome. It will work with Firefox just as fine. And you can type in training.ensembl.org. Zoom in a little bit so it's a bit bigger for you. And at the top of the page, there is a link that says training materials. So if you could just click that link, and all of our materials are in this first link here. So the one that says webinar. On this page, you can find my slides on the right-hand side. So this is just a PDF version of what I'm going to talk about. But at the bottom here, under demos and exercises, these are actually quite useful. So these are walkthroughs for the live demonstration I'm going to do towards the end of the webinar. So if I've gone through an example too quickly, you can always ask me to repeat it, or you can look at the walkthrough in screenshots in your own time on the training page. All right, so let's switch back to my presentation. So all of these materials as well, they are going to be available to you indefinitely. So even maybe in 1 week's time, you've forgotten how to retrieve, for example, variation, you can always come back to that website and go through the walkthrough in your own time. Okay, so let's begin. So first of all, I want to talk about the Human Pangenome Reference Consortium. So introduce you to this project. So we humans share about 99.9% of our genomes, and the remaining 0.1% is really what makes the human species so diverse. And it's what contributes to our different physical appearances and also health profiles. Populations of different ancestries can have genomic variations that are specific to their population group. So for example, we know that the ability to digest lactose into adulthood is a trait that varies a lot among populations. The lactase persistence allele, for example, is quite common in populations from Northern Europe, which have a very long history of cattle domestication. But in comparison, the trait is much less common in East Asian populations, where dairy isn't really a traditional part of the diet. So for example, I am lactose intolerant, so I have to take lactase tablets to actually digest the lactose myself. So the current human reference genome, the current human reference genomes, sorry, there are multiple, and in general, the genomic data are predominantly of European ancestry. But we know that no single population can represent the diversity of the entire human species. So using a single reference genome creates a reference bias. It can negatively impact the variant discovery and also gene disease association studies and the accuracy of genetic analyses. And this is why the announcement of the first draft first draft results from the Human Pangenome Reference Consortium was so big. So in May 2023, articles were popping up all over, from high impact journals like Nature and Science to institute announcements like the National Institutes of Health, NIH, to mainstream media like CNN and New York Times. I'm sure you've seen a couple of these notifications on your phone. And all of these articles highlighted the importance of a human pangenome. So scientists need a range of reference sequences that include genomic between, so that diverse populations are better represented, and not just populations of European ancestry. The Human Pangenome Reference Consortium, or HPRC, is a huge collaboration between major institutes across the Western Hemisphere, one of which is Ensembl's European Bioinformatics Institute, of which Ensembl is part of. And the goal of the HPRC is to produce a pangenome from a cohort of genetically diverse individuals. So the aim is to generate high quality assemblies for at least 350 individuals, so 700 haplotypes. And the first 200 samples, or the first 200 assemblies, will be coming from cell lines from the 1000 Genomes project. So this was a project that cataloged many common human genetic variants across different populations. A more diverse human reference genome can improve our understanding of the genetic variation in different human populations. And this can in turn help researchers develop and inform clinicians with a more personalized prediction, diagnosis, and even treatment of various different health issues. So on my slide, I've got a little graph of the number of individuals that have been sequenced so far, and the populations on the left-hand side. So far, the HPRC has produced genome assemblies of 47 individuals, or 94 haplotypes, from 16 different populations. So these populations are from all across the Americas, Africa, and Asia. Okay, so that was a little introduction to the HPRC project. So now I want to talk about the HPRC data in Ensembl itself. So for those who are unfamiliar, Ensembl was launched in 1999, and the initial project goal was to automatically annotate the very first draft of the human genome, and also integrate it with other data and make it publicly available. Ensembl maps annotations and different genetic features, so for example, genes and genetic variants to genomic regions, which gives the genomic region value. And throughout the years, more and more genomes have been released and added to Ensembl. And today we actually have several Ensembl sites. So you can see an overview of the different sites on my slide here. All of the sites follow a very similar layout. So once you're familiar with, let's say, the main Ensembl site, you'll have no problem navigating any of the other sites. So the Ensembl main browser is dedicated to vertebrate species. So this was the very first browser that was published. And it also includes some non-vertebrate model organisms. The Ensembl Genomes browser is dedicated to non-vertebrate genomes, and it is divided into different taxa. So each taxon would have its own kind of color coding, but the layout will still remain the same. Both of these browsers, both the Ensembl main browser and the Ensembl Genomes browser, are updated about every 3 to 4 months. We also have the Ensembl Rapid Release browser, which is a more lightweight genome browser compared to the Ensembl Genome site. So this was launched in 2020 to allow for the faster release of the latest genome annotation for large numbers of vertebrate and non-vertebrate species from various sequencing projects, like the Darwin Tree of Life. We also have a couple more specific portals. So we have the Ensembl GRCH37 portal. This only includes the older human GRCH37 assembly and genomic data. And we also have a separate portal for the coronavirus genome. One thing I forgot to mention here, the Ensembl Rapid Release website. So in comparison to the Ensembl main and genome site, this is why the data there is much more lightweight, as we are updating it quite quickly back to back. So you can access all available HPRC genomes through the Ensembl Rapid Release browser. If you want an overview of the available genomes, you can go to this very first link here. Don't worry, I'm going to show you during the demonstration how this looks like. Basically, just a table of all of the different individuals from the HPRC project. You can also access the data directly on the Ensembl Rapid Release website. So that's the second link here. And if you want to download any whole genome data, you can also access all of the HPRC data via our XGP site. So in Ensembl, we've got all 47 assemblies available. For each individual or each sample, both the maternal and paternal haplotype assemblies are available. You can also access the annotated T2T genome in rapid release if you are interested. And I also want to highlight that all Ensembl data is completely open source, open access. So this means that you can use Ensembl data in your own research completely free. But if you do use the HPRC data, please don't forget to cite publication. I do have a slide of both publications towards the end of my presentation as well. Okay, so let's talk about the different types of genetic data that we have for the HPRC genomes. So the annotation, we have gene annotation data, and the annotation for all haplotypes were generated by mapping gene annotation from the GRCH38 reference to each haploid assembly. So this included a subset of the GenCode 38 gene set. So the GenCode project, for those who are unfamiliar with it, aims to produce a high quality gene set in human and mouse as well, based on biological evidence. And it is also used as the standard reference gene set in major projects like the 100,000 Genomes project by Genomics England. So then we mapped the genes to the corresponding target genome by looking at the surrounding regions of the genes. So by locating the most likely corresponding regions. And we also performed pairwise alignment of the reference, so GRCH38, and the target, so the pangenome regions, to map features of genes, so for example, exons and intronic regions. We also excluded read-through genes and genes on patches or haplotypes from the GenCode gene set. So this is why this is only a subset, because we have excluded some of the genes. Variation data is also available for the pangenomes. So for this, we lifted over GRCH38 variation data from both Nomad and ClinVar using CrossMap. And we predicted the consequences, the variant consequences by Ensembl, by Ensembl's variant effect predictor, or VEP. There's also VEP caches available for the genomes. And then finally, we also have comparative genomics data available, although this is being expanded as we speak, actually. So we are working on expanding this to include gene trees and ortholog-paralog relationship predictions as well. But currently, we have homologs of closely related and model organisms available for the annotated genes. And these were aligned using Diamond. Okay, so I can see that you are asking loads of questions in the background. Thank you guys so much. Thank you, Alina, Ben, and Francesca for answering them in the background. So what I'm going to do is I'm going to move on and do a demonstration of the HPRC data in Ensembl Rapid Release. And for this, we are actually going to look at two different sites. We're going to look at the Ensembl project site and the Ensembl Rapid Release site. So I'm going to begin with the Ensembl project site. So let me move to my browser, and I'm going to open a new tab. And I'm going to type in projects.ensembl.org. So projects.ensembl.org, and then I will hit enter. And what this page is showing you are all the different projects that use Ensembl code in the research. So I mentioned the Darwin Tree of Life, and what we're focusing on today is the Human Pangenome Reference Consortium. But please feel free to explore all of the other projects on this page after the webinar. So let's click on Human Pangenome Reference Consortium. So that's just the second link here on the left. And I will zoom in a little bit. So this is the page for the HPRC data in Ensembl. So we've got a short description of the data, and underneath we've got a list of all of the assemblies that we've got available at the moment in Ensembl. So this is data from 47 individuals, and we give you both the paternal haplotype, so PAT, and the maternal haplotype, MAT. In the table, you can find more information about each of the samples or the individuals. So we've got the assembly name, the population, so which geographic location they're from or they were based in, the accession ID, so this is the sequence ID, who submitted the original sequence, annotation files. So you can get the whole genome annotation in either GTF or GFF format. You can get the FASTA for the proteins of the whole genome, transcripts, variants, and other data. So these would be the homologs of the genes within each of the assemblies. If you want to view the actual data, the actual genomic data in Ensembl, there is a link at the very end of the table that says rapid.ensembl.org. So if you click that link, so let's click the link for this very first individual here, that will lead you to the page for the individual we just clicked, the species page, species information page. So at the top, we can see this is the species, Homo sapiens. We've got the sequence ID and the assembly name. Let me just click this pop-up window away. And if you scroll down on the right-hand side, we've got some summary statistics. So we've got information about who submitted the assembly, when it was submitted, the length of the genome, when the gene annotation was released, number of genes, and so on. So all the summary statistics on the right-hand side. On the left-hand side, you can find the available data for this assembly. So for this assembly, we've got gene annotation data and variation data available. You can actually click on or view some examples. So for each type of data, datum, I think it's actually for singular, you can click on example gene, and that will lead you to an example gene for this assembly. So this is one way you can find the available HPRC data in Ensembl. But if you are already within the Rapid Release website, you can find it within Rapid Release as well. So let me open a new tab and pretend like I didn't do all of that in the Ensembl project site. And let's say I'm starting off at rapid.ensembl.org. So if you type in rapid.ensembl.org in your browser, this will lead you to the Ensembl Rapid Release website. So this is updated about every 2 weeks. And if you scroll down, we've got the section called All Genomes, and you can click on View and Download Available Data for all species. So if you click that link, that will lead you to the list of species that are currently available in Rapid Release. So all of the species available in Rapid Release. The first thing you'll notice in the table is you can actually show more than 25 entries. So you can change the number to show 10, 25, 50, 100, or even all of the species. But I just want to see all of the Homo sapiens assemblies that we have available. So I'm going to use the filter in the top right-hand corner. I'm going to type in Homo sapiens. And as I'm typing it in, you'll actually notice that it will filter down our table to only include Homo sapiens. So if we now scroll down, we can see that our table should only contain Homo sapiens. At the very bottom, you can see how many entries this applies to. So it's only showing 25 of 97 entries. So what you can then do is scroll back up to the top of the table and change it to all. So select the drop-down menu and select all. And now, if you scroll down the table again, you'll notice that this table is now much bigger. So this will help you see or have a better overview of the different HPRC genomes we have available here. You can also download the table. So there's this little Excel icon at the top right-hand corner. So you can download what you see. So this would be your filtered table as you're seeing it right now. Or you can download the whole table. So all of the genomes available in Rapid Release. Sometimes you have some hidden columns as well. So if you click Show Hide Columns at the top of the table, there are a few columns that are hidden. So for example, genome size, assembly name, and so on. So you can unhide them by clicking on the column name. One thing you can do with the table as well is you can sort it by column. So let's say you want to sort all of your Homo sapiens by common name. So you can just click on the column name, and that will filter the table alphabetically. Okay, so this table is actually quite similar to the table we saw in the Ensembl project page, with just a bit more information in there. So in this case, we also have the annotation method. These links that I'm highlighting with my mouse, so these are the links that will lead you to the FTP site to download the whole genome data files. But what it will tell you is the type of data that is available for each of the assemblies. So for example, for this second Homo sapiens assembly, so this is a maternal haplotype of this individual. We've got gene annotation data available. We've got the entire genome available and homology data. So let's click on this very second example, not very first, the second example, and find more information about the species itself. So we've already explored this. We've got the summary statistics here on the right-hand side, and what type of data is available on the left-hand side. You'll notice under genome assembly here, so on the left, the second box, there is a drop-down menu for all other human assemblies. So you can, if you click the drop-down menu, you'll actually see all of the different human assemblies, so all the pangenomes that we have available in Ensembl. But it's a bit difficult to find your, it can be a bit difficult to find your genome of interest in here. That's why I like to use the all species table that I just showed you earlier. Okay, so if you want to find information about a gene within this assembly, you can use the search bar at the top of the page. So let's say we want to look at information of the HBB gene in this individual, in this maternal haplotype of this individual. So I'm going to click, type in HBB and click go. And that will lead me to the results page, or search results page, sorry. So sometimes you'll have multiple entries for HBB or multiple entries for your results. And this is because Ensembl will give you all of the entries that mention the term HBB in it. In this case, in our case, there's only one entry for this, and this is the gene, so hemoglobin subunit beta. And this is exactly what I'm looking for. So I'm going to click on the gene ID to open the gene tab. So while this is loading, you'll notice at the top of the page, we have two tabs open. We've got the location tab. So this is where you can visualize the HBB gene graphically in the genomic region. And we have this gene tab. So this gene tab will include any gene-related information. For our case, we've searched for HBB. So by default, you land on the summary page. So that's what you're seeing here on the right-hand side. We've got the description, synonyms, so what other names this gene is known as, the location on this assembly, the reference equivalent. So this would be the entry in the GRCH38 assembly in the reference. So let's just click on this and see how this looks like. So that will lead you to the Ensembl main site for the GRCH38 reference for the HBB gene. So if you wanted to compare this gene, the gene in the HPRC individual with the reference sequence, for example, you can jump between pages that way. I'm back on Ensembl Rapid Race now, back on the HBB tab. So if you scroll down, we've got more information about this gene. We can see that there are 6 transcripts for this gene. So a gene in Ensembl is described as a set of transcripts or splice variants or isoforms. So if you open this table, we will give you each of the transcripts that this gene is transcribed to. So that's what you're seeing here in this table. Let's just hide it for now. Scrolling down even more, you can find some more information. So there is a link, an external link to the HGNC symbol, the Ensembl version, the gene type. This gene is protein coding, and the annotation method. Scrolling down even more, you can see the region and detail view. So this is what you would see on the location tab, but this is just more of an overview rather than in detail. So our gene or the transcripts of our gene of interest are highlighted in this green color. So that's what I'm highlighting with my mouse right here. So we can see we have 1, 2, 3, 4, 5, 6 transcripts. All right. So in this tab, you can find a ton of information about HBB. For example, you can find information about all of the variants that have been or that are known. Sorry, all of the variants found within this gene. So on the left-hand side, in the gene-based displays in this panel here, you can click on variant table. And this would change your view on the right. So now, instead of summary information, you're seeing a table of all of the variants found within HBB. So this table is very similar to the all species table that we looked at earlier. So we've got the information for each variant per row. We've got the name, details, location, the type of variant, the source. So remember, these are lifted over from GRCH38, from NOMAD and ClinVar. Evidence, clinical significance, consequence type. So this is the sequence ontology term. So the consequence of the variant on the gene. And if it's protein coded, if it's a synonymous or non-synonymous variant, we've got the amino acid change as well, and the transcript that it affects. You can also filter this table, so you can filter by column. All you need to do is select. So in this case, I've selected consequences. You can turn all of your consequences off. Let's say you only want to view missense variant. So what you'll need to do is search for the missense tag in the list, click apply, and that will reload your table. And if you look under consequence type, so this column towards the right of the table, we can see that we're only now displaying missense variants. So this is one way to filter down variants within HBB. For those who want to download the entire table, there are several ways to do so. You can download the table again using this Excel icon just in the top right-hand corner of the table itself, or you can export the data itself. So here on the left-hand side, just underneath the gene-based displays, there is a button that says export data, which will give you a pop-up menu of the different formats that you can select. So the different formats you want to download your data in. So for example, if you go to the output option, you can see you can download the variants in bed format. You can download the FASTA sequence, You can download the annotation in CSV, comma-separated format, GTF, GFF, and so on. So depending on what your downstream analysis is going to be, you might need to select a very specific format. You can change some options. So if you want to show some flanking sequences as well, you can change that here. And all you need to do then is click next, and that will download it straight to your local machine. I also want to quickly show you the homologs display. So I'm back on the gene tab, and I'm going to click on homologs here on the left-hand side under gene-based displays. So this display will give you all of the homologs from a set number of species. So these can be closely related species or some model organisms. I'm just waiting for it to load right here. I hope my internet is not giving up on me. So let me just, every time I want to change my tab, it loads up. So this is perfect. So the table here gives you the reference species. So that's what I'm highlighting in the first column. The name or the gene name in the reference, the stable ID. So this would be the ID again in the reference. So for the first example, it would be chimp. The type, you can always hover over anything that is underlined like this, and it will give you a description of what the abbreviation means, or just a description of what data we're showing you, the identity, and the coverage as well. So again, just like before, you can download this table as you see it, or you can, you can download the table as you filtered it, or you can download the entire table. But it's not a big table here. So we've got for our homologies for this HBB gene in this individual, we've got the model organisms, human. So this would be the HGRCH38 reference, mouse, and so on. All right. So the final thing I want to show you, let me just check the time. The final thing I want to show you is the location tab. So if you want to visualize genes in the genomic region, also graphically, you want to switch to the location tab. So I've clicked on the location tab. You can see this now highlighted. By default, you land on the region in detail view. So that's what you're seeing here on the right. And this is actually the genomic region displayed in 3 different resolutions. So we've got the genomic region in the chromosome overview. We've got it in the region overview, so in the 1 megabase overview, showing you the region of the gene. So again, highlighted in green. And 100 megabases up and downstream of the gene, so to see it in context with other genes. And if you scroll to the bottom of the page, this is what we call the region in detail view. So just like the name says, it's just the gene, but in more detail. So instead of seeing the gene, we're actually seeing the individual transcripts on here. So that's what's highlighted in this green color again. And in this view, you can actually add and remove tracks. Unfortunately, this is still being built. So the current tracks that you can turn on and off are the variance tracks here that are on right now. But as time goes, we'll be adding more tracks to this view. So right now, what you're seeing here, so what I forgot to mention is what you're seeing here are genomic features mapped to the genome, and these genomic features are represented in what we call tracks, genomic tracks, genome tracks. So we've got a genes track here. So this is showing genes, or in this case, transcripts mapped to the genomic region. We have variants from NOMAD. So again, each of these individual lines or boxes represent a single variant from NOMAD mapped to the genomic region. And we've got variants from ClinVar as well. And if you scroll down, underneath, you can find a legend of each of the colors and what they mean. But the nice thing about this view is you can actually export the image. So if you want to visualize something in a report or in a publication, you can export this image. So click on the export image just on the top of the view. This is available for each of the views. So if you click export image for the region in detail view, it will download the region in detail. If you export image for the region overview, it will download this overview. So you just need to select which resolution you want. So all you need to do is click on export image. That will give you a pop-up window again, where you can select the format. PDF is usually a very good format. The resolution is very good. And then all you need to do is click on download. You can also export the data. So again, if you want to export the annotation data of the genomic region, you can do so by clicking on export data here on the left-hand side, or you can even share this page with your colleagues. So let's say you want to highlight a specific region in the genome. So you can actually change your mouse action to either drag or select. I want to select a region. You can click and drag your mouse and highlight or mark that region. And let's say you want to send this to your colleague. So all you need to do is just click on the share button, this share button here at the top of the region in detail view, or this share button in the left-hand side. That will generate a URL, and that's the URL you can then send to your colleagues. If you are hoping to just copy the URL up here from your browser, your colleagues might not see the same configurations as you. So for example, I've actually added this, I've actually highlighted this region in the genome. So if I were to copy the URL from my browser, my colleagues wouldn't be able to see my marked region. So I always recommend for you to share or to generate a URL using this button if you want to share a page with your colleagues. Okay. So there's much more to explore for the data in HPV, but I think I'm going to stop there. I just wanted to give you an overview of how you can get started on exploring data from the different HPRC assemblies. So let me switch back to my browser, to my slides. And before I take questions, or before we move on to the Q&A, I just want to finish up with some closing remarks. So I hope that this webinar gave you an introduction of the HPRC project and also how Ensembl is involved in it. So the HPRC aims to produce a pangenome from a cohort of genetically diverse individuals. And this will provide a better representation of the human genome, an improved understanding of genomics as well, because you'll have a more diverse set of pangenomes, and a fair, an equitable way of precision medicine. So a fair way to develop personalized medicine even in the future for all populations. So Ensembl specifically maps genomic features to genome sequences, and we also perform our own analyses on the data. And with regards to the HPRC data, you can find this in the Ensembl Rapid Release website, which integrates gene annotation, variation, and homology data for the currently 47 individuals available from the HPRC project. If you enjoyed this webinar, we also, the outreach team, the Ensembl outreach team also provide courses that are more tailored to exploring the data on the browser, the Ensembl browser. We also have a more advanced REST API course, so how to access Ensembl data via our REST API. And we also offer train-the-trainer courses. And if you are interested in learning more about these courses at all, please don't hesitate to email us at helpdesk@ensembl.org, or you can find more information about hosting a training workshop on the training website. There are different ways to reach out to us. So we are on social media. We are on Facebook, LinkedIn, and X. And we also have a blog where we publish any updates, for example, to Ensembl. If you need any help, or if you want to learn more about Ensembl or bioinformatics in general, really, the EBI training team have a very good portal here where you can do courses in your own time online for free. We also have Ensembl-specific tutorials on the ensembl.org website. And if you want to get in touch with us, again, social media, or you can email the helpdesk@ensembl.org address, and you can also sign up to our mailing list. So I mentioned if you're using Ensembl data or HPRC data, please do cite the most recent papers. So for Ensembl, this would be Ensembl 2024, and the latest or the HPRC paper that you would cite for the 47 currently available assemblies is this paper here, a draft Human Pangenome Reference by Liao et al. And then finally, I've used the word we a lot. It's not we at all. It's these beautiful people who work on the data. So Ensembl is not just Alina, Ben, Francesca, and me, of course. We're actually a team of about 100 people, and that's just an Ensembl alone. So it takes a big team effort to keep this database running and keep it relevant to researchers around the world.
Thank you very much. Thanks, Louise, for today's webinar, and thanks everyone for joining in. And thanks to Francesca, Ben, and Alina from Ensembl team to join this webinar and help with the questions.
So I can see that there is an incoming question, and it's quite a technical one. So the question is, I've made a genome assembly with many sequences of 80% divergence. After making a graph, some fragments are not linked. Could you recommend me some tools or techniques to modify the graph again? So I think this is actually a really good question for Francesca, because it sounds like quite a technical one. So I don't actually know the answer. So I'll let you take that, Francesca.
Okay, thanks. Yeah, so I have to say that in Ensembl we provided mostly the annotation. So let's say the first part, the graph is something that the other colleague in the consortium are dealing with. So just to say that I'm not a graph expert, as I tried to reply to the previous answer that it might happen that if there are there is a lot of divergency, it might become difficult building the graph, and some fragments are not linked. It's a question of understanding how important are these fragments according to the rest of the graph, and it's a question of finding a balance in the entire pangenome, because going too much in details, it means customizing the parameters in the tools. It means taking those sequences, but it will become more difficult making the graph generalized. So it doesn't mean that the graph needs to contain everything, but it will be helpful having a general graph. So you need to find a balance and play a bit with the parameters. And about the tools, yeah, as I said, the tools that you use are the right one, the one that you cited in the previous question. About the specific parameters, my best advice, it's what I usually do, is talking with the developers, because they usually test these tools so much in details, and they might be more helpful in this specific situation.
Thank you so much, Francesca.
Any comments on gene annotations in the regions identified as gaps in HG38, but not in T2T?
So this is again quite a technical question. I'm not sure if Francesca, you have any comments for this at all. Did you work directly with the T2T genome yourself? The annotation for the T2T.
Well, I have to say that we know that the 38 is not complete. So the question now, and I hope that the pangenome and building the graph will help in that, will be dealing with these kind of gaps, because we obviously the T2T should be the most completed. So now the interesting part is to compare the gaps that we have, understand why do we have them, and projecting the 38, that is the reference to the other target, working on the, let's say, unmapped region. So what in 38 is a gaps might be found in the other, and we want to identify and work on this kind of region in order to obviously improve the reference.
Thank you so much.